some of the parts of his talk are going to be featured on skeletal stem cells and our current research that revolves around this topic. And there's a few reasons why we study skeletal stem cells. But first of all, it helps us to understand how we are shaped the way we are, unlike the other organ system that we study, the hematopoietic system. Bones are not amorphous, and they have helped define the ability of the vertebrates to occupy many distinct niches across the animal kingdom. And so a large part of that is the ability and the versatility of these skeletal stem cells to form different types of matrices. Furthermore, as we age, the structure of the bones overall change. And so there is a question of whether or not this change is perhaps reversible, and whether or not we can find factors that we can help maintain the skeleton in its ideal functional and structural form. And there's a lot of genetic questions as to what are the key players, what are the major signals, and when do the key events of how the skeleton forms takes place and how it does that. And last but not least, one of the key topics in our lab that relates to skeletal stem cells are how do different types of stem cells talk to each other. And so this is an important topic for us because many different types of tissues are organized together within organs. And these tissues, as their organism grows, they require a new source of cells. And the source of these cells ultimately are the stem cells. And organs tend to be relatively complex. And so there has to be a high degree of coordination between the activities of the different types of stem cells within the organs. But very little is known about how that actually takes place. So my introduction into stem cells and different types of stem cells coordinating their activity together in a complex organ system began when I joined the lab of Irv Weissman, who now more than 30 years ago with the Herzenbergs and Jerry Spangler discovered the first known tissue-specific stem cells. And this is the hematopoietic stem cell, also known as a blood-forming stem cell. And since that time, Weissman and his colleagues have not only identified the self-renewing hematopoietic stem cells, but also characterized how it gives rise to the complex cells that constitute the immune system and also the mature blood lineages. So this is a diagram of the so-called hematopoietic tree. And each of these cell types are intermediates through which the hematopoietic stem cells gives rise and differentiates into very, very diverse cell types, from macrophages to T cells to B cells,
no, thank you.
to red blood cells, etc. And here are the intermediates, and it's important that these intermediates were isolated, and these arrows define the lineage relationship, because by solving this lineage relationships and by subsequently comparing these cells genetically, we get to learn about the genetic processes and also the signaling events that determines how such a pluripotent, multipotent, undifferentiated cell like the hematopoietic stem cells can give rise to specific types of mature lineages. And what is listed also here are the cytokine switches that can control whether or not a hematopoietic stem cells eventually becomes an erythrocyte, a red blood forming cell, or mature T cells. You know, it would have to follow these lines of differentiation along with these and also exposure to these various types of cytokines. And when I joined the lab, my question was, where do these signals come from? Now, the mature sites of hematopoiesis in adult land-dwelling vertebrates resides in the bone marrow cavity. And this is a relatively complex tissue. Here's a cross-section of what the bone marrow cavity looks like. And you can appreciate that it's made of many different cell types. There's these round globules here. These are fat cells. Here you can see that there are a line of red blood cell containing vessel cells. This yellow staining region here are osteoblasts. And there's various fibroblast types and nervous tissue that's also all within this type of, within the bone marrow cavity. And somehow these various different cell types are supposed to signal to one another to help maintain the process of bone formation, as well as blood formation. So our question is, how do we find the different types of stem cells that resides within the bone marrow? And our approach for doing this is using the technique of flow cytometry. As probably most of you know, this is a picture of one of the more recent models of a flow cytometer. And you can, and for those of you who haven't been exposed to FACS before, it's just simply a glorified high throughput jelly bean sorter. The way it works is that it allows us to label cells with different colored antibodies that sticks to unique proteins that are expressed on the cell. And so these are fluorescent conjugated antibodies. And by passing it through this flow cytometer, the computer can pick up the signals, and it would be represented as a plot like this. So on the X and Y axis represents two different types of surface molecule that's found on a candidate cell, and also the relative intensity of the events that's detected by the cytometer that of cells that expresses these signals. So for instance, in the red square here, these are going to be cells that are positive for both Thai and also this marker CD105. After the computer detects these signals, it turns on electromagnets, and then it allows the various cell droplets to be sorted into individual collecting tubes or wells. And from these specifically sorted cells, this is what we can use to analyze the function of specific populations in bone marrow or any other types of tissues. And so one way that we can ask whether or not a particular population is the stem cell of a particular type of tissue is by transplanting it. And so here is the overall overview of the assay that we have developed to identify unique stem cell types that resides within bones. So our first finding was that the adult bones tend to be relatively more difficult in terms of isolating stem cell types. And possibly it's because it's also at that time full of very dense matrices. And so we found that actually it was a lot easier to isolate stem and progenitor cells from fetal bones. So then we were able to establish a technique for mechanically and enzymatically dissociating fetal bone cells into single cell suspension. We could then apply surface antibodies that labels the surface proteins expressed on these cells and separate them into different types of populations, which we would then embed in a matrix to secure the cells in one location. And then we would transplant them. And the site that we transplanted these cells in is the kidney capsule. And the reason why we selected the kidney capsule, it's because it has this dense membrane that allows you to transfer cells within that's underneath this membrane forms a sort of an envelope, and the cells would be directly exposed to the vessel-rich kidney parenchyma. And that helps to keep whatever cells that are there viable, and they tend to quickly vascularize. So we were able to find a population of cells that gave rise to blood vessels. So these are blood vessel progenitors. These are green tubes that are filled with red blood. And then we found another population of cells that gives rise to green adipocytes, so adipocyte progenitor. And lastly, we found a population of cells that when transplanted into the kidney gave rise to an ectopic piece of bone. And very excitingly for us, when you were to look at, so this is another picture of these ectopic bones that are formed from GFP labeled progenitors, fetal bone progenitors that are transplanted to the kidney. And if you were to take a cross section of this bones, you can appreciate that there is actually a marrow cavity that has formed within this ectopic bones that's growing now on the surface of the kidney. And here is a CT scan showing this ectopic bone. You can also make out a little bit of a marrow cavity there that's adjacent to the vertebra. And so within these ectopic bones, we also find stem cells of the hematopoietic system that stains for their characteristic markers SCA and KIT. And not only do these stem cells appear to be hematopoietic stem cells, but they're actually bona fide functional hematopoietic stem cells because they have the ability to rescue lethally irradiated mice in a transplant setting, and also has the ability to give rise to all the mature blood lineages and immune lineages that a hematopoietic stem cell normally produces. So we have found this progenitor population that makes bones, but at the same time, it now also makes a home or niche for a completely different type of stem cell, the hematopoietic stem cells. And so this allowed us to perform experiments to understand some of the signaling and the genetic processes that are important for this process of niche formation or hematopoietic regulation by these bone stem progenitor populations. And so if you were to knock down this gene Osterix in these cells, it inhibits their ability to form this marrow cavity, and yet the bones still remain. So that kind of shows how the different stem cells within an organ system, such as the bones, are tied to one another. We were looking for the cells that regulates the hematopoietic stem cells, but that actually led us to a completely other different new type of stem cells. This is the skeletal stem cells. Now, to be honest, when we first demonstrated that fetal bone progenitor populations could give rise to an ectopic bone in a transplant setting and also support hematopoiesis, we weren't actually sure that there were a skeletal stem cell there. So I had the great fortune to be mentored by Mike Longacre for my postdoc studies. And so that was the question that I was trying to address while I was in his lab, was whether or not these bone progenitors actually constitute a new type of stem cell. So using a genetic mouse system that labels every single cell in a mouse a different color, we call this the rainbow mouse, you can track stem cell activity in situ. And what I mean by that is stem cells have the ability to self-renew and give rise to other cells. But if they don't migrate very far from one another, they tend to form a colony. And so we know that bones are actually composed of just more than just bone tissue. They also can compose of cartilage tissue. For instance, in this central picture here, this is a cross-section of a femur, and the yellow stains for bones and the blue stains for cartilage. And this is the growth plate of a femur. And this is the growth plates of a femur from a rainbow mouse. And so you can see that this region here that has bone tissue and also cartilage, green staining cartilage tissue, is actually composed of clonal individually labeled columns of skeletal colonies, so that you can see that there are some pink colonies here adjacent to some blue colonies. So this in situ multipotent colony formation is a telltale sign of a tissue-specific stem cell. And so since these types of activity reside within this area of the bone known as the growth plates, we then micro-dissected out each of these different regions and compared them by FACS. And we were able to find that there were these combinations of surface markers that allows us to split these bone populations into 8 different populations. So we, again, utilized the renal capsule transplant technique to evaluate the function of these cells. And so here you see a pentachrome staining of a cross-section of a femur, of a mouse femur. And this is a relatively immature mouse, so that's why there's a lot more cartilage, which is stained here in blue. And there's also some bones, which is stained here in yellow. But the individual populations that we were able to resolve using these panels of surface markers gave us one population that only gives rise to blue staining cartilage, 4 that gives rise to bones, and 3 that gives rise to bone marrow. So one of our questions was, are these representative of individual stem cells, for instance, the cartilage-specific stem cells, or bone-specific stem cells, or bone marrow-forming stem cells? Are there 8 different types of stem cell populations within the bones? Or is it perhaps possible that these are actually related to one another? And so to make a long story short, by transplanting each of these different populations and then afterwards reharvesting them and seeing what they turned into, we were able to establish that these 8 populations of cells were actually intermediates of one another. And they're related to each other by this lineage map that we had established. So similar to the hematopoietic system, at the very peak of this lineage map is a self-renewing skeletal stem cell. It has the ability to give rise to downstream progenitors, but importantly also has the ability to make more of itself. And furthermore, at this stage, which we call the bone cartilage stromal progenitor, or BCSP, this is where it splits off into distinct cartilage-specific, bone-specific, and stromal-specific progenitor populations. And so as Mike mentioned, we did this in 2015. And using this as a guide, we were also able to now identify the human skeletal stem cells fairly recently. But this was a little bit difficult at first, because the first thing that we realized was that the surface markers from mouse skeletal stem cells were completely different from those of the human tissues. And so, Mike and I discussed this, and the solution that Mike came up with and taught me was something that I haven't actually learned before. It was called Sutton's Law. And I've never heard of a famous scientist named Sutton, and this is a picture of Will Sutton. But it turns out he's no ordinary scientist. Actually, his expertise is in breaking into banks. So he's a bank robber. And so the advice that Will Sutton gave his trainees was, go where their money is. And so following this advice, we asked, well, since we found that skeletal stem cells in mice resides at the growth plates, perhaps the human skeletal stem cells are also within the growth plates. So we then micro dissected out the the growth plate regions from human fetal bones. And then we're using a technique called single cell RNA sequencing, which allows us to compare the transcriptome. So the transcriptome, unlike the genome, is basically all of the expressed genes within a cell. So we found that there were cells within the growth plates of humans that were quite similar to mice, and they shared a majority of the expressed genes. And so there were some surface markers that were unique to humans that we were able to identify was expressed in this population. And by testing these markers, we were then able to, as in mice, identify specific bone-forming populations, specific cartilage-forming populations, and populations that contain bone, cartilage, and stromal cells, or generate tissues that contain these tissues after you transplant them. And so they were organized there. One of these is actually the stem cell that gives rise to all of these other cell types. And not only that, we were able to show that it has the ability to give rise to itself, and it has both primary, secondary, and tertiary colony formation ability, and also the ability to give rise to other types of skeletal tissues. So it has the ability to self-renew and also is multipotent. This marker for human skeletal stem cell allows us to identify skeletal stem cells not only in fetal tissue, but also in adult, iPSC-derived. These are the famous reprogrammed induced pluripotent stem cells that one could program from the skin using the famous Yamanaka factors. And also in the case of ectopic skeletal genesis that can be induced by a very powerful morphogen, BMP2, when applied in a very concentrated solution under the skin. So having identified now the skeletal stem cells in mice and also in humans, we can then use this to understand how the skeletal stem cells is relevant to skeletal regeneration and skeletal diseases. And I, with Mike's help, was very lucky to recruit Owen Marisik, who is now a medical student here at Stanford, to work on this project. And it turns out that Owen has a specific expertise in breaking bones because he was also part of their football team and was also part of the 49ers and played professional football at some point. And so Owen devised this fracture assay that we still use in the lab, whereby we can break a mouse femur in half and yet have it stabilized with a pin. During this time, the broken bone would form a piece of regenerative tissue called a regenerative callus. And we were able to find that within this regenerative callus was our phenotypic skeletal stem cells. So as this callus size increases, then the frequency of the skeletal stem cells also increases. What was also unusual was that when Owen transplanted and compared the activity of these fractured skeletal stem cells to adult skeletal stem cells, he found that normally resting adult skeletal stem cells only made a small piece of bones when transplanted to kidney. This is unlike newborn skeletal stem cells. If you transplant an equal number, they make a fairly large piece of bones with cartilage and a marrow cavity, but this is the small slivers of bones that you would get by transplanting an equal number of adult skeletal stem cells. So it looks like the adult stem cells has already lost some of its skeletogenic activity. But if you transplant an equal number of fractured skeletal stem cells, you can see that they have actually very similar bone forming ability as the newborn. So this also corresponds to expression of genes that were shut off when the newborn mice or became more mature. So somehow, at least in the case of bones, what doesn't kill you can also make you younger, because this injury event has somehow triggered re-expression of fetal developmental genes. And this also corresponds to fetal or very youthful levels of tissue forming ability. So this same injury-related amplification of skeletal stem cells was also observed in human tissues. And we were able to establish a model for looking at how skeletal genesis takes place in humans. With this xenograft system, we would transplant these fetal limb tissue isolated from human fetuses, many of which has actually been aborted because of medical reasons. And then we would transplant them under the skin of newborn immunodeficient mice. Very interestingly, these bones actually grew into, once they vascularized, into a much larger bone. They would follow the scale of the development in human tissues rather than in mice. So there's this very large finger that has now grown in the back of the mouse, and it contains joints and also marrow cavity. And if you were to introduce fractures within these bones, you would also see a corresponding increase in the human skeletal stem cells, as we have observed in the mouse system. So by being able to identify the human skeletal stem cells, this gives us the ability to look at various types of diseases from the perspective of cells that are most important in forming those types of tissues. So one thing that, one of the major killers for people over the age of 75 are falls. And so it seems that for whatever reasons, broken bones in the elderly tends to heal much less efficiently. They're also more fragile than younger bones. And so it's not really understood completely whether or not stem cells play a role in this efficient type of healing. So postdoc in their group, in collaboration with surgeons in the orthopedics department and also the plastics department, was able to analyze tissue specimens collected from patients who had undergone procedures related to fractures. And so they were able to look at two different cases. In the case of complete regeneration, the bones actually tend to form relatively well. But then in the case of non-unions, the bones are unable to repair themselves and form the necessary amounts of bone tissue within the normal time frame. So a student in the lab is looking at why this occurs and found that in the elderly, actually, although there were still relatively high frequencies of the stem cells, they curiously lose the ability to form bones. These are showing individuals. So we actually collected 400 individual patients' skeletal stem cells. And then we performed functional assays for each of them. And these are actually alizarin red stains that compares the osteogenic activity of skeletal stem cells from different patients. For instance, here we're looking at an 82-year-old versus a 59-year-old or a 21-year-old. And the red is actually positive for bone formation. And the white is because although there are cells there, these cells have somehow taken on a fibroblastic form and are unable to ossify. So it seems that as one of the consequences of aging, at least in the skeletal stem cells, is that the skeletal stem cell compartment becomes occupied by variants of these stem cells that are skewed in terms of their tissue formation towards fibroblasts, but not bones. So now with Malakia, we're now looking at the genetic processes that controls why these skeletal stem cells tend to be fibroblast skewed rather than bone or cartilage skewed. And we're hoping to identify genetic mechanisms or in their signaling that can help us reverse these phenotypes. So this approach was used by a former trainee of ours, Ruth Tevlin, who I had the great fortune to work with. And we applied the stem cell lens to looking at a very similar problem in diabetic patients or diabetic mice who also suffer from poor fracture healing. And by comparing purified skeletal stem cells even though diabetes is a very complex disease and there are many types of, various different types of tissues are actually affected in this systemic disease, we were able to, by comparing normal skeletal stem cells versus diabetic skeletal stem cells, we were able to identify a specific signaling pathways that diabetic skeletal stem cells were deficient in. And this is the Indian hedgehog protein, which is normally expressed by skeletal stem cells in a fracture setting, but due to the upregulated inflammatory state that's present in the diabetic circulation, TNF-alpha actually shuts down Indian hedgehog production by injured skeletal stem cells. And so very fortunately for us, our neighbors are actually experts in hedgehog signaling, and they were able to provide us with recombinant proteins and also small molecule agonists that when applied to skeletal stem cells in a diabetic mice was able to reverse this effect. So this is looking at skeletal stem cells from a diabetic mice now treated with Indian hedgehog. And you can appreciate that it has restored its ability to form bones. And it's also corresponds to the rescue of the mechanical strength of the healed bones to a level that resembles normal wild type non-diabetic mice. So by isolating the skeletal stem cells, we're able to use this as a lens to understand different types of skeletal diseases. And so the previous few stories I've told was about one of the types of tissues that the skeletal stem cell produces, and that's bone tissue. But the skeletal stem cell also makes cartilage tissue. And so we were also able to find that by controlling the ability of skeletal stem cells to form bones versus fibroblasts versus cartilage, we could actually also apply the same lens towards understanding osteoarthritis and why cartilage isn't able to repair itself in adults. And this work was done by a very talented team in the lab, Matthew Murphy, who was a visiting medical scholar from Ireland, and Lauren Kopke, who was a previous CERM scholar, but now a technician in the lab who is applying to graduate school in the fall. And so Matthew and Lauren first, again, used this rainbow mice that, again, allows us to label every single cell in a mouse of different color. And then we asked whether or not we can track clonal cartilage formation in the joints of developing mice. And here is a picture of the rainbow cross-section of a newborn mouse at postnatal day 3. And you can appreciate that there's a lot of different color clones on the chondral surface. But at 2 weeks, there's already starting to be less clones and fewer still at 6 weeks. So that by 1 year old into a mouse's life, which is probably equivalent to around 50 years in in terms of a human lifespan, there's hardly any clonogenic cartilage formation left. And so this lack of clonal formation in adults explains why there's much reduced cartilage formation and why cartilage isn't able to regenerate itself. And also it suggests that at the cellular level, that there aren't any actual skeletal stem cell left in adults. But because of the previous finding from Owen and Marisik that injuries can induce local skeletal stem cell amplification, Matthew decided to test a relatively well understood surgical technique called microfracture, which is commonly used to repair cartilage defects in younger individuals. And what this technique involves is using a specific drill. The surgeon would punch holes within the exposed chondral bones to access the underlying marrow cavity. A blood clot would then form, and this clot would be replaced by what is called fibrocartilage. Now, although it's called fibrocartilage, it's not actually cartilage. It's more of a type of fibrous scar tissue that covers this exposed bone. But it's better than having just bone rubbing against bones. But unfortunately, this fibrocartilage doesn't really have the same elasticity or the same mechanical properties as normal cartilage. So it tends to wear away over time. So Matthew then devised a technique to study microfracture in mice. And he was able to observe that what happens in microfracture in mice, at least, is that you again see this amplification of skeletal stem cells from injury, but at a more localized setting. However, what happens is that these injured regions tend to form a variety of tissues. Some of them has the ability to form cartilage, but many of these clones actually give rise to fibroblasts, and also some gave rise to bones. And so it was already a revelation for us that using this surgical technique, you can use it to initiate localized wound healing related signaling that can induce local stem cells to amplify. However, it seems that although these amplified stem cells can have the ability to give rise to different types of mature tissues, they require additional signaling to make the right type of tissue. And so by previously solving this lineage map of how the skeletal stem cells gives rise to bone and cartilage, we've actually conducted comparative gene expression analysis among these various subsets and found that BMP2 is capable of amplifying these skeletal stem cells, but that there was an additional downstream signal, VEGF, which is normally known to be important in blood vessel formation. And actually, drugs that targets VEGF signaling, Avastin, is commonly used to treat cancer because it's a way to cut off the blood supply of the cancer. Somehow, however, when you apply VEGF if you block VEGF signaling in developing bones that's produced by skeletal stem cells, you completely inhibit its ability to give rise to bone or fibroblastic stromal cells. So instead, if you block VEGF signaling, all of the skeletal stem cells then turns into cartilage. And this is what we observed in vivo. So here we have introduced these relatively large defects on the chondral surface of a mouse femur. And here we have applied a hydrogel. This is 4 weeks after transplant of a hydrogel containing just PBS versus another hydrogel that contains BMP2 and VEGF inhibitor, and one that contains BMP2. And what you notice is in the case of the hydrogel containing PBS and BMP2, you have formation of either white fibrous tissue or yellow staining bones, but not cartilage. But you do see cartilage if you were to apply these two signals together, BMP2 and soluble VEGF and this VEGF blocker. And so this blue staining large chunk here is actually a piece of stable articular cartilage. So these cartilage, if for anyone in the audience who is actually familiar with the bone field, so the process of bone formation actually normally involves a cartilage intermediate called hypertrophic cartilage. Although it has properties similar to cartilage, is actually not relatively not stable, and eventually most of the hypertrophic cartilage turns into bones. And so hypertrophic cartilage is actually marked by expression of this molecule, collagen 10. And so you can see in the PBS setting, the control, basically the regenerated material forms a lot of collagen 10 positive tissues, and also with BMP2. But unlike these instances, if you were to combine BMP2 with VEGF blockade, then you form a piece of cartilage that is positive for all the mature markers known to be associated with hyaline cartilage, which is like aggrecan, collagen 1 and collagen 2, but not collagen 10, and not another marker for hypertrophic chondrocyte, which is MMP13. So this is 8 weeks post microfracture, and the effect is durable. And so the cartilage persists for up to 16 weeks post microfracture. And so that's 4 months in a mouse's life, but it's equivalent to at least 25 years in a person's life. So this stable cartilage that is formed also has the same mechanical properties as regular and normal cartilage. And so we were able to measure the mechanical properties of these regenerated cartilage using atomic force microscopy. And this just shows that there's very little difference between the regenerated cartilage versus uninjured cartilage in terms of their Young's modulus, which is a measure of its elasticity. However, if you were to look at the tissues that were regenerated with just BMP2, you can see that it's actually a lot higher. It's a lot stiffer. So it looks like cartilage, it feels like cartilage, but it doesn't behave like cartilage. And so in order to test whether or not it's able to restore function to these mice, we were also found a facility at Stanford which helped us conduct these so-called gait analysis. And so this is actually a screen that a mouse runs on top of, and it can record the way that each paw is landing. And so we can compute its gait analysis. And so what it shows is that the cartilage forming factors, BMP2 and soluble VEGF, actually allowed us to rescue the normal gait of a mouse much better than the other factors. And also it corresponds to a reduction in pain. So this is as measured by the grimace test. And so when mice are under a certain discomfort, you can tell because they bulge their cheeks or they narrow their eyes. So the BMP VEGF combination dramatically reduced the pain in these treated mice. So although the previous studies show that this combination BMP2 and soluble VEGF inhibitor actually can generate cartilage in mice, we still weren't sure whether it would be able to do the same thing in human bones. And so therefore we tested this in our xenograft model, whereby we transplanted these fetal bones in mice and showed that indeed this same combination of factors, BMP2 and also VEGF inhibitor, gives rise to stable cartilage in human tissues. And what is also very exciting from a translational point of view is that both BMP2 and VEGF antagonists are already FDA approved. And so one could envision potentially if we can find a proper combination and the right type of scaffold, we would be able to, in principle, bring this rapidly to the clinics. And so it was by virtue of this ability to show that, you know, we have been able to regenerate a tissue that has shown very little regenerative capacity before. The public, it was also picked up by the New York Times, and so folks were very excited about this, and also Forbes magazine. So in summary, by looking for the origin of a type of stem cell that supports blood, this eventually led us to identifying a new type of stem cell, the skeletal stem cell, which we show can be found in a variety of bone sources, including bone, ectopic bones, adult bones, and also iPSC-induced. These stem cells can respond to local injury cues brought upon by application of surgical techniques. And these activated skeletal stem cells can then be directed towards forming the right types of tissues using specific factors. And although we were able to establish that this type of approach works for regenerating a skeletal tissue cartilage, I think it can also be anticipated that other types of tissues that seemingly have a very reduced regenerative ability in adults, for instance, the heart, might potentially also contain resting stem cells that could be induced to expand or become activated by local surgical cues, and then be directed to forming the right types of tissues using the appropriate signals. So, so just to go into a little bit about the follow-up work that our group is conducting. So we're now trying to understand in greater depth how this process of local injury can stimulate regeneration. So how can surgery stimulate regeneration? And so these are some of the new critters that we have in the lab. This is an axolotl. It's a type of salamander. And it is the grand champion in the vertebrate world in terms of regeneration. And so, although we were able to show that cartilage can regenerate itself, all you have to do to demonstrate the axolotl's ability to regenerate is to actually amputate an entire limb. And this surgical procedure can actually trigger such a pronounced regenerative process that it can regenerate not just a type of tissue, but an entire limb. And it's not restricted to limbs. It can also regenerate part of its brains, its heart, most of its liver, etc. And these type of surgery related injury cues are also important for potentially understanding the usage of surgery for treating cancer, because it's also known, although it's very poorly understood, as to why surgery can sometimes become a trigger for distant metastasis. So we're thinking that it's partly due to the fact that there are many different types of stem cells in a particular tissues, and they coordinate their activities with one another in a type of social network. So that if you were to block one of their processes or eliminate one of these types of cells, right, it would actually lead to a dramatic change in their tissues. For instance, if you were to block vascularization of bones, the entire bones would turn actually into blue staining cartilage. if you were to block blood formation in a bone, then the marrow cavity instead would become full of fat and not bones. So there's this whole interdependent processes that's occurring at the single cell levels in tissue within the stem cell microenvironments. And I'm running a little low on time now, so I don't have a lot of time to go into in depth, but basically we have developed a new method to conduct single cell spatial transcriptomics of these microenvironments. We have a process to isolate out chunks of tissue that had been isolated, for instance, from the edges of a surgery of a fresh wound. And then we have a method to colorimetrically label every single cell. And then take these cells apart for single cell RNA sequencing, which gives us the most comprehensive view of the various genes that are expressed in the individual cell, and thus gives us a window into understanding the signaling through which, so this is a cluster of cells, and we can basically, using this approach, figure out how the genes within each cells are changing and respond to a certain event, such as a local microfracture injury. And so these are the types of colorimetric cells. These are the clusters of tissues that we can label colorimetrically that we are mapping using microscopy. And then we have an algorithm that allows us to break apart each of these individual cells for single cell RNA sequencing. And then we can piece everything back together again. And that gives us kind of like a comprehensive view, but at the single cell level, how the different signaling is taking place. And so we have talked about using these signals and delivering them using scaffolds as a way to modify the microenvironment and controlling the fate of stem cells. But another approach that we're starting to develop is also taking advantage of a cell type that we frequently find are in injured tissues, and that is actually a type of blood cell, monocytes. So we're developing a genetic approach to modify monocytes so that rather than having them express factors that are used for normally a tissue clearance, we can now ask them to make regenerative factors such as BMP2 or other molecules related to stem cell activity. And so I just wanted to thank this great team that that are in the lab right now. And so these are some of the students and fellows who have worked on this project. And I am very well supported here by a vast network of very experienced PIs. And these are my funding sources.
Thank you, Chuck. That was quite a tour de force.